So I am moving this over. So today I'm going to tell you a story. So I'm going to tell you a story that started in 2016, where we had this vision of using feedback control to program the fate of cells. And in order to get started, I am just going to give you a brief overview of what I'm going to talk about. I'm going to talk about pluripotent stem cell reprogramming, which was the beginning of our vision, and then formulating reprogramming as a robust control design problem. And from there, how do you actually implement these robust controllers in cells to achieve your end goal? And then I'll show you our recent results on using these controllers in action for reprogramming cell fate. So let me get started with background on stem cell reprogramming. I always start with showing the Waddington epigenetic landscape, which is a metaphor to depict the process of cellular differentiation. So we can imagine a pluripotent stem cells is at the top of the hill as it's the highest potential. And then at the bottom of the hills that are more highly differentiated cells in different lineages. And going up the hill, the differentiation potential increases. So it was long viewed as a unidirectional process, differentiation, until in 2006, there was a remarkable discovery from Yamanaka and colleagues that demonstrated that you can reprogram the fate of a fibroblast, which is terminally differentiated cells, to pluripotency by overexpression of a bunch of key transcription factors. These are transcription factors that in their initial cocktail included these 4 key factors, OCT4, SOX2, KLF4, and CMYC. And what they saw is that by delivering this to the cell in high amounts, some cells could turn back into pluripotent stem cells. And so at a high level, what are we going to talk about today? So that at a high level, you can regard this process as an open loop control problem where you have artificial transcription factor overexpression is a constant input to your system. You can almost regard it as a control input. And then the output is the cell phenotype that you're trying to control. And in between, right, the input and the output, there is the whole complexity of the cellular circuitry that it's actually, right, dictating what the fate of the cell is. So in this talk, I'll try to close the loop around this and have genetic feedback control circuits inside the cell to steer the concentrations of transcription factors towards optimal levels. So why do we care about stem cells, right, or induced pluripotent stem cells? It's because if we have them and they come from a patient, we can then use them to regenerate, right, the tissues that the patient may need due to injury or sickness. So in the long term, and this is a very long-term vision, right, you can think of using these for engineering living materials for tissue regeneration, or also for reprogrammed patient-specific cells. And there is a lot that is thought of right now, right, in terms of extracting cells, reprogramming them, and differentiating them, for example, to beta cells. As we know, diabetes is a very diffuse problem, especially in this country. So what's a little bit of numbers that we should keep in mind is that this reverse process of taking a fibroblast to a pluripotent stem cell, it's possible, but its efficiency is extremely low. So it's 0.27%. And that means that it's not really practical for everybody use right in the clinics. What people know very well is that the levels of transcription factors, especially the levels of these OCT4 transcription factors, are important for the success of this process. And there are a number of additional studies that focused in details on how the level of OCT4 defines the entry or the exit from pluripotency. So bottom line, the summary from these slides is that the OCT4 transcription factor is probably the most important factor in this cocktail at least. And it's known that the defined level, it's important for pluripotency. So for us, since you are trying to solve a control problem, it's becoming clear from this picture that OCT4 is going to be our actuator, okay? So now the next question is, this is biology. How do we try to translate biology into a more formal control problem that we can try to tackle? So at a high level, right, we have the cell, and the way in which reprogramming works is through overexpression. So you encode the gene that you want to express at high levels into some synthetic DNA that I will refer to as genetic device moving on. And then you deliver it to the cell such that it's becoming chromosomally integrated. There are many ways to do that. Lentiviruses, it's one way to do that. But mathematically, right, what you can think of is to describe the rate of change of this key transcription factor in the cell, right, which is supposedly driving the fate of the cell. And of course, who is driving the dynamics of these transcription factors? Well, there are what I'm depicting here, Hill functions, which come actually from the gene regulatory network that is controlling OCT4 among other factors. There is, of course, decay and degradation. And then there is this external input that you are applying through this genetic device, which is the overexpression. So traditionally, this input UI is an overexpression, so it can be regarded as a constant production rate. So within this view, then you take your reprogramming problem in the context of a dynamical system where you have your function here with the Hill function, the decay, and your overexpression, and you are asking if by overexpressing, so adding this input, you can move the state of your cell, okay, from one basin of attraction to the other, thinking that each basin is reflecting a different cell fate. For example, this S0 could be your pluripotent target cell state. This could be, for example, the endoderm state. This could be the trophectoderm state. Each basin of attraction is essentially regarded as the set of states that lead, right, to that final fate. So when you apply overexpression, what happens to this landscape? So this landscape has three basins of attraction, just as an illustration. Once you create overexpression, so you're adding this constant input, the landscape changes. And the idea is that you will be able to create an equilibrium point that is asymptotically stable, to which your trajectory will converge, so that when you remove the input, you have converged there, and then you stay in that basin of attraction, right, of S0. When you remove the input, therefore, you have successfully, right, reprogrammed your state to the target pluripotent state. So we will say that the GRN, so the gene regulatory network, is strongly reprogrammable to your target state if there is an input such that the resulting attracting set of this input is contained in the region of attraction of your target set. So that means that if you apply this input, essentially it doesn't matter where you start, you're going to land in the region of attraction of your end state here. So now I want to point out a reflection here that there are some limitations to constant overexpression. So in particular, we have a fact here that has a proof, but I'm not going to the proof here, that a cooperative gene regulatory network, so a network where you have all essentially activation among all the nodes, is strongly reprogrammable only to its maximal stable steady state. So if you look at the core pluripotency G regulatory network, you have OCT4, SOX2, and NANOG. They are all in this positive feedback loops. And so this is clearly a cooperative network. Also, you know that the pluripotent state by literature has intermediate OX4 level and intermediate SOX2 level. So it's definitely not maximal, right, in the space of concentrations of transcription factors. So therefore, according to this simple analysis, it looks like constant overexpression may not actually be able to let you reach this intermediate state that easily. So then you can start looking at the weakening the property of reprogrammability. You can ask to weakly reprogram to the state as 0 from an initial condition if there is an input such that the attracting set of X naught is contained in the region of attraction of S0. But if S0 is not maximal, there is no guarantee, essentially, that a cooperative GRNL is weakly reprogrammable to it. So these all big reasoning is just to say that for the core pluripotency GRNL, constant of expression is not necessarily being able to lead you to your pluripotent state. So you need better control than constant inputs. So this is just an expression to show you what happens if you take a model that we have developed many years ago of this core gene regulatory network that controls pluripotency. This is an example of a two-dimensional reduction of how the nucleins work. You have 3 equilibria. The intermediate one here is the pluripotent state. And so upon addition of your input, what happens is that this pluripotent state disappears almost right away. So that means that if you start from either S1 or S2, you won't be able to reach that state by overexpression. This can be also seen by what are called bifurcation diagrams, where you can put on the x-axis here the strength of your overexpression. As you increase your overexpression strength, essentially your intermediate state, which is what you need to reach, disappears right away. So if you start from any other state, it's really not possible unless you have noise in the system, but that's a different story, to essentially land on the state here. So with that, that prompts the question on, okay, so how do we try to solve this problem in a way that there is some guarantee, okay? So if we regard essentially the gene regulatory network as a bounded disturbance, okay, so we're taking this big network there and we are assuming it's just disturbing, right, the level of transcription factors that we are trying to control, then our input can take more interesting forms. For example, you could try using integral control. So where the input UI is essentially the integral of the error between the current level of transcription factor and the final level. Or you can try to use high gain feedback, okay? But bottom line, if you use this and you plot here your XI concentration with this one being the target, well, you're guaranteed, right, that if your gain G here is sufficiently high and here as long as K is positive, that you're going to converge to these XI here. So with that, then the next question is, how do you implement, right, this control loss on this overexpression? So there are at high level 2 ways, right, in which you can implement these controllers. One is inside the cell. So you find a way to create your biomolecular reactions such that UI is given by this control loss. And in essence, what happens is that every cell receives a different input based on its local state, which is different from a computer-based control where all cells receive the same input based on the population state. So the second approach is not viable for this problem because we have a high level of cell-cell variability, and therefore it's not desirable to have the same inputs applied to all cells. Rather, each cell should get the input reflecting its internal state. Now, this is the stage at which we were in 2016. There was no biomolecular controller available at the time for robust set point regulation in mammalian cells. So this is then going to a major undertaking that went into the design of biomolecular controllers inside the cells for robustness, specifically for mammalian cells. So what's the approach here? So again, we are going to regard, right, our system here that we are trying to control as our genetic device. And so what happens is that the production of this transcription factor, OX4, for example, is going to be subject by a number of disturbances. Of course, there is the gene regulatory network, right, that is perturbing it in all possible ways, because actually it's what controlling that level. There is stochasticity, there is cell variability, there is the cellular environment. When the cell fate changes, there are many changes that happen in this intracellular environment. The resource level is changing. There is reallocation of transcription factors to different processes. So it's major changes that are uncontrolled, right, and unmeasured. So those are disturbances. So these disturbances can be regarded as unwanted and unmeasured inputs on your plant, right, which is the gene expression process that can affect either translation or transcription. In particular, for transcription, we definitely have the effect of the endogenous GRNet, gene regulatory network. This is controlling the promoter of OX4. We have cell state changes, we have changes in transcription resources, we have changes in chromatin state. All of that is a big set of perturbations on your system. And so how do we ensure that the output Xi is robust to disturbances? So here is a temporal plot that it's tracking with respect to the publication in SynBio, the publications in Control and SynBio, which have been increasing at a similar pace just because, right, today we realize that a lot of the problems we encounter are often due to lack of robustness, right, to perturbations that you have in the cell. And something interesting, as I'm dashing here, is that a lot of the work on control design mammalian cells really started to appear after 2018. Okay, so it's much more recent than what happens in bacteria. But when we had this vision was 2016. So that means that there were 2-3 years of gap, and that's the gap that we felt had to fill with finding these controllers. So we therefore undertook this task of having the following problem solved. We have a genetic module that takes inputs, among which transcription factors. We have outputs, proteins of interest, and then we have all these disturbances. How do you fix that? Well, you can either use a negative feedback control approach, where you take the output, right, of your genetic module, and you use to feed a feedback controller that then adjusts variables, right, in your genetic modules, probably rates. The second approach is feed-forward control, where you have a way to indirectly measure the effect of the disturbance on your output so that you can compensate, right, for it directly on the output. Bottom line, doesn't matter which one of these two you use, if they're designed correctly. This is what you should see on the left-hand side plot. When the disturbance appears, the output, instead of responding to the disturbance and leaving your desired set point, it's actually adapting to that disturbance and reaching back the set point. So with that, then I'll go to how we implemented negative feedback control, in particular integral control, and then we'll move to feed-forward control. So what's the setup here? Again, we have inputs V that we can modulate and disturbances D that we cannot touch. And we want the output, right, to stay at some desired reference value. So we are going to use a quasi-integral controller. So integral control, as you may know, is exerting an action on your system, which is our genetic module, which is proportional to the integral of the error between the reference value, which here I'm denoting by V, and the true output, okay? Under stability of the system, this control law will ensure that your output will be completely independent of D at steady state if your reference value is constant and also the final value of the disturbance is constant. Although there are results that hold for time varying disturbances and input as long as they change slowly. So here the challenge to integral control was that molecular decay is unavoidable in vivo due to cell growth and no specific degradation. So this led to integrated leakiness, which I'm depicting here. And the outcome of that is you want to find a way to make this leakiness small, right, in comparison to the control action. So that's what we call quasi-integral control, which is you are making through a gain here, 1 over epsilon, with epsilon very small, the integral part much larger than the decay part. And of course, this is the paper by Ely, who is actually here, who demonstrated that under certain conditions, right, you can actually have perfect disturbance rejection using this strategy as epsilon goes to 0. So, which is essentially what's being said here. So bottom line, if we think of biomolecular implementation, what this is telling us is that we need controller reactions that are fast to capture this 1 over epsilon parameter and compute the difference between the reference and the true output and integrate that difference. So we worked on several implementations. This is one of the implementations that we built in mammalian cells. So what you see on the right-hand side is a cartoon of this quasi-integral controller that is using phosphorylation cycles or even covalent modifications of proteins as the key process to implement this integral. So the way it works is that you have your output protein Y, which indirectly it's causing the dephosphorylation of X star. X star is an activator of the protein itself. So essentially here you have a negative feedback loop. On the other hand, the input is triggering the phosphorylation to X star, which then drives, right, the promoter output. We should also remember where all these disturbances happen. They happen along this path, right, of gene expression. So if you write the differential equation for this system that is just using the standard Michaelis-Menten functions for phosphorylation and dephosphorylation, and then protein expression, and then you assume first that you can make your phosphorylation cycle work in the so-called zero-order regime, where essentially the substrates are exceeding the Michaelis-Menten constant, so that essentially this term and this term are saturated. And you have timescale separation, so you have this parameter epsilon that is very small. It's the ratio between the decay rate here, proportional to dilution, and the rate of the phosphorylation reactions, which typically is much faster. Then you have this form on the right-hand side, and if you call V the reference input, the ratio of K1, K2, which are the two catalytic rates of the two modifications, then you see the quasi-integral control structure. And your quasi-integral variable, it's Z, which is the concentration of X star. So stability is ensured if X star is much smaller than KD, which is the binding constant of the promoter here. So theoretically, what this is saying is that while in the open loop system where this feedback is disabled, the open loop is going to suffer from the presence of the disturbance. In the closed loop system, the two should be on top of each other. So with that, we are going to go now to the implementation here. And this is, as I am citing up here, this is the work by Ross Jones, who is here in collaboration with Healy Hyun. So Ross built a system. We have the output of interest, which is a blue protein here. What we did, we 2A linked this blue protein with a phosphatase that essentially dephosphorylates the on-part protein, which when bound to the VP64 activation domain, it's activating the output. So there are several components in the system, so I'm going to describe them one by one. So the input of the system is this engineered activator protein. So OnPAR is a protein from bacteria and belongs to two-component signaling systems in bacteria. So the OnPAR-MZ pair is a very well-studied two-component systems in bacteria. So what Ross did, he essentially verified, first of all, that OnPAR can bind to its operator sites, right, even in mammalian cells, by checking that you can essentially have expression, in this case of a minimal CMV promoter, when you fuse OnPAR to an activation domain, in this case VP64. So the OnPAR sequence allows you to bind to the promoter, the VP64, it's the activation domain that allows you to recruit the transcriptional machinery, including the mediator complex, RNA polymerases, and all of that to activate gene expression. There was a lot of work that was done in the system because the MZ, in quotes, kinase is a bifunctional enzyme. So it was working both as a kinase and both as a phosphatase. So after several rounds of mutations, Ross was able to decouple the two functionalities, so we could get an almost pure kinase that phosphorylates the protein and almost pure phosphatase, right, that dephosphorylates the protein. So this is really where this integral feedback X star is happening. Now, the disturbances that we tested this controller against are two. So we had resource sequestration, which was engineered via expression of a potent activator here that in prior work was shown to sequester a bunch of transcriptional co-activator. So this is essentially inhibiting this expression right here. And then we also engineer microRNAs that could target directly the transcript here to see if we could be robust to them as well. So in the sake of length, I will just show the results for the disturbance applied by GAL4 VPR. And on the left-hand side here, we see the open loop left and the closed loop right. So in the open loop, we see the response of the output, the blue protein, to the input kinase with and without this disturbance. And you can see there's a large difference there. On the other hand, in the closed loop system, we have essentially no substantial difference between the two. And the response, as you can see, is much more linear-like. Moreover, if you look at the noise between the open loop and the closed loop system, you can further see that the closed loop system is able to keep a much more concentrated distribution. So it's decreasing the coefficient of variation of the output as it's expected by negative feedback control. So that's for the negative feedback control. So we also engineered feed-forward controllers in the cell, which I'm going to show next, essentially with the same idea to ensure that at this point an output constant, in this case, would be robust to perturbations. So I'm comparing here an open loop or unregulated system where we have a bunch of disturbances hitting your transcription translation process, and the regulated system in which we engineered the feed-forward control by an ERN, which is essentially a protein that binds a target sequence on the mRNA and targets that for degradation. So we engineer a bunch of controller, either using ERN or we did also with microRNAs, but I'll just focus on the ERN side. So the key idea of the feed-forward controller is that you have these two paths, assuming these two promoters are identical, that are subject to exactly the same disturbances here, so that you have the disturbance affecting similarly, right, your output mRNA and the level of your ERN, so that once you look at the steady state, essentially you have that these disturbances cancel out, right, on your final output, assuming that the catalytic rate, right, of your ERN is sufficiently high, for sure, compared to dilution. So in this case, the expected behavior is that compared to the unregulated system, which is suffering once you are increasing change in the disturbance, the ideal system should have no change, and the regulated system we expect to be close to the ideal. So going back to applying our disturbances to the transcription rate of the system, we again expressed GAF4 VPR, which is sequestering transcriptional resources from the two transcriptional modules here. And what we saw comparing the unregulated system and the regulated system, we see that the unregulated system had a substantial decrease in the output fluorescence as you keep sequestering resources from your transcription, while the regulated system essentially remain at the same level. And this was also published by the PhD work, right, by Ross Jones and Healy Hyun, were both here. I should also mention that if you look at noise, this feed-forward controller is also able to quench noise. This is noise mostly due to cell-cell variability coming from DNA copy number variability, since all of this is with transiently transfected right, circuits on plasmids. So for our reprogramming application, something that was particularly interesting was to check if this feed-forward architecture could actually keep consistent gene expression level across different cell types. And so what we did, we then transfected the circuit, either regulated or unregulated, to different cell types. And what we saw in this plot is that while in the unregulated system, as you can see here, there was quite a bit of difference in the expression level of your output across different cell types. In the regulated system, they are essentially almost all lying on the diagonal, meaning that different cell types are essentially keeping very similar expression levels. So the bottom line conclusion here is that feed-forward control to some extent also insulates gene expression from this variable, right, intracellular environment on which you don't have any control. Okay, so all of this takes us back to where we started, which is the problem of reprogramming. So going back to this picture, okay, so at this point, we are in 2021, more or less. We have some tools that we have developed, and now we can revisit this picture, okay? In revisiting this picture, we have to think about, once we insert our genetic device in the cell, how are we going to do that? There are many ways. One way that is, or at least was in 2020, when we really dabbled into this problem, a commonly used method is lentiviral delivery. So you essentially encode your circuit in lentiviruses and you affect the cells. So then the virus makes your circuit integrate in the chromosome. The challenge with these is that for lentiviral design, you need to pick the parts such that they don't require much space in terms of length of base pairs numbers, okay, on your gene, because there is a limited cargo. And I will say that for many of these applications, that's one big limitation. And in fact, there are a lot of researchers that are doing very important research on figuring out, okay, what delivery method can we engineer that allows larger cargos to get into cells. So, but bottom line, then we thought that we need to remove all the lengthy parts in our design. So the ultimate designs that we landed on are all based on RNA, okay, and specifically microRNAs. So going back to our picture here, we have our endogenous gene regulatory network, which is controlling our OCT4 transcription factor. We are going to encode, right, OCT4 by a construct that is expressing OCT4. And so if we do that and we write the total OCT4 in the cells, we'll see that it's going to be determined by the endogenous network, which is here, by my overexpression, and I'm putting this big G to denote that is really high, okay? I'm just flooding the cell with this OCT4 right now. And we have some perturbations, okay, on the DNA copy number, because with lentiviral delivery, we don't know how many copies are going to get in. Now, we want to reach some robustness to all these red perturbations. So the idea is to implement a controller via this microRNA that is going to accomplish two functions. One is it's going to knock down the endogenous OCT4 so that it doesn't contribute much to your total OCT4. And then it's also going to increase the degradation rate of your target mRNA so that here you can essentially implement what turns out to be a high gain negative feedback. So if you write the differential equations of the mRNA that you're trying to control, you have disturbances here, here, you have a high gain feedback component that comes from this large microRNA that is targeting both of this. And you also have a feed forward term here where this microRNA being expressed by the same DNA as your target, it's essentially filtering out the perturbations coming from DNA copy number. So this is a very compact way of combining a high gain feedback with a feed forward control. So you can do a little bit of math. The system is asymptotically stable, but bottom line, if this gain G is large enough, then your mRNA level for your OCT4 is going to become essentially independent of your endogenous GRN and of the perturbations that affect DNA copy number. The final thing is tuning. How do you tune the level of this OCT4? Because we said there is an optimal level. From the expression that you see here, there are some parameters that are tunable. This K parameter, it's hinging on the catalytic rate of the microRNA, on which you have some control by changing essentially the base pairing between the microRNA sequence and the target on the mRNA. So here is some simulations that are showing that as you increase this gain G and you have any target OCT4 level, it doesn't matter if your endogenous GRN is pushing your system to be in this low OCT4 value, then your system is going to be able, if G is large enough, to regulate the OCT4 level around any value that you want, theoretically. And so how do we use this in practice? So we have two steps here. So we first essentially use the controller to identify OCT4 trajectories that are conducive to reprogramming. And as a second step, we use this controller to enforce the OCT4 levels identified in step one. So for step one, so this is more of an exploratory task, right? So we are using the controller as a discovery tool. So what we want to do to span as much trajectories as possible. So we generate a highly variable set of trajectory by essentially removing the feed-forward controller here because now we want all this variability in levels to come in effect, because we want to explore, right, a high, potentially a very large trajectory space. And then we exploit amplification of intrinsic noise due to low molecular counts, okay? So we essentially use this TRE promoter, which is controlled by this RTTA activator. If this RTTA activator is in very low molecular counts, then the noise at the transcription level is going to be highly amplified. And these are just known results. Then what we did, we tracked the single cell OCT4 trajectories via microscopy and then classify them as successful or not by performing Tra160 staining, which is a marker of pluripotency at the end of the process. So this is just to show what we ended up using for very low RTTA dosages. We get almost the full spectrum, okay, of amnion green levels here. So this is the process. The process takes 21 days. So we infected our cells with the viruses containing the circuit, and then we performed daily imaging up to essentially 21 days. So this is just an example of the images. There was a lot of work to actually make OCT4 and neon green nuclearly localized so that we could essentially see this very nice compact nuclei here. And then there was some analysis that show high correlation between the actual OCT4 level, right, across the cells and the M neon green levels. So we know that therefore neon green is a good proxy for OCT4 in order to identify the OCT4 trajectories. I also want to point out that it's critical to have the microRNA targeting endogenous here, because we do not want to have the endogenous contributing to the OCT4 level that controls the cell fate, because we can only measure what neon green mirrors, which is what we ectopically express. And so this is just showing data that with this microRNA, we are essentially knocking down the endogenous OCT4 level throughout the reprogramming process, so that whatever we measure here with neon green is actually the total cellular OCT4 level. So this is our process. We performed daily imaging for a number of days. And at the end, we essentially classified colonies in 3 types. Type 3, which are positive essentially for Tra160, are what's called the bona fide iPSCs, which is what we want. Type 1 and type 2 are still pluripotent stem cell colonies, although they are not considered the top quality colonies. So this is just a movie. So these are wells in which you see our controller in actions while these colonies are getting formed. And so this is the time course of 21 days. So we took trajectories of each single of these cells and compiled them on temporal trajectories for type 1, 2, and 3 colonies. Something that is immediately popping up from this is that you can form a colony only if your OCT4 level, which is what essentially is depicted on the y-axis, is keeping a constant value, which is at this optimal value here that you see in these trajectories. Any cell that actually goes below, right, that is not going to form any colonies. So cells that maintain stably higher OCT4 level led to iPSC colonies. Cells that could not keep this level all along did not generate colonies. Interestingly, only 30% of the generated colonies are type 3 iPSCs, which is saying that, yes, this constant OCT4 level throughout the process is necessary, but alone it's insufficient to generate the best quality of iPSCs. So if you look in terms of OCT4 distributions on the x-axis, what I'm showing you here is the distribution of the whole, across the whole population of cell. The type 3 colonies require distribution that is essentially here, which is very narrow, and it's hitting in this beam. So at this point, then, the summary is that this is the Y distribution that we use so far, but success, right, requires for your cells level to be around this distribution here. So what we did, then we used this controller to enforce supraphysiological for levels identified in this step. And we used for this the constitutive promoter to reduce intrinsic noise here. We also put back the feed-forward control in order to mitigate noise coming from DNA copy number variability. Okay, so we are trying to squeeze this distribution as much as possible. And then we varied the level of the distribution, so the mean of the distribution, by varying the affinity of the microRNA to the target site, which is here. So we therefore had 4 controller variants with different levels of this affinity here, which is changing the set point value. And these are the experimental OCT4 distributions that we obtained. So these orange ones are for the 4 variants. And as you can see, compared to what we wanted for type 3 colonies, the V4 version is the one that best captures what we think the best value of OCT4 is. So what we did, we therefore took all these 4 versions, we reprogrammed the cells through the same process, and then stained and counted colonies for each of these variants to see if indeed the V4 resulted in the best outcome. So this is the data. So looking at here, what we see is that V1, V2, and V3, even if they're just slightly different, they resulted in very little colonies. However, V4 definitely outperformed all the other ones, and also the reported lentiviral efficiency from the literature by about twofolds. So this is essentially a first indication that robust control of transcription factor levels can improve, right, reprogramming outcomes, mostly by taking more of your cell to express at the levels that you want. So the summary of this part is that we use the controller as discovery tool and discover some new biology. But on the other hand, we found that using controller to enforce optimal OCT4 level improves efficiency, but it's still not what would be expected from theory, okay? So from theory, right, if we were able to enforce this level, right, then we would obtain 30% efficiency based on our trajectory tracking here, but that's not the case. So why is that happening? So this is just some debugging that we did to see why we didn't reach 30%. So the cells with higher OCT4 levels double less frequently. So that means that the cell distribution over time is taken over by lower expressors that grow faster. So what it means in terms of temporal trajectories that yes, we start at the optimal level, but slowly we drift to lower level. So we are not actually keeping that constant level all the way through. So that's ongoing work. So if we want to hope to reach this 30%, it's necessary to mitigate the influence of OCT4 expression on doubling time, which is reported also in the literature. So summary, so we recasted the problem of reprogramming as a dynamical system problem and a control system problem. We engineered biomolecular feedback controllers for mammalian cells application to have robust control over transcription factor levels. And we combine feedback with feed-forward control to robustly regulate OCT4 level for iPS pluripotent stem cell reprogramming. Next, so going along this route, there are 2 things that we aren't controlling for, which are the big suspects here. The first, as I said, is the effect of OCT4 expression on doubling rate. And the second, we haven't talked at all in this talk about chromatin state, but it's playing a big role. And that's actually what my research is currently focusing on. So this is our team that I want to acknowledge. So all the reprogramming experiments were done by our collaborators at Harvard. Thorsten Schlerer and Catherine Ilya has been instrumental, especially in the last part of the project, to get those reprogramming experiments running. And then the theory, it's Ely, see here, that all the theory that was here is essentially done mostly by Ely. And then Ross covered a critical role in the design, idealization, right, and construction and experimentation of all the biomolecular feedback controllers. And then Nika Shakiba also had major roles in the development of the microRNA controllers. And that's about that. I'll stop here and take any questions.